Brine shrimp were used as a model organism in this experiment due to their resiliency and adaptability in harsh environments. This is important because observing these shrimp can further our understanding of biotoxicity and in turn be applied to other species. The hypothesis of this experiment was that the 0.4% toxicity variation of the nanoparticles would gravitate toward and eventually stick to the shrimp. The nanoparticles were also predicted to cause a change in the shrimp's physical characteristics and eventually kill the shrimp due to the toxicity concentration of each solution. To test this hypothesis, 5 test wells that contained different concentrations of nanoparticles in seawater were used as environments for the shrimp. Each well was labeled according to the concentration. The ratios of seawater to nanoparticles included 0, 1 to 1, 1 to 10, 1 to 100, and 1 to 1,000. After the test wells were loaded with the different concentrations, 10 brine shrimp were placed in each well. The brine shrimp were then observed periodically over the next few days in order to record observations. Our assay was a 0.4% 20 mg per milliliter stock solution. Well 1 was the control group, which contained no nanoparticles. It was pure seawater. Well 2 contained the 1-to-1 dilution, which was 1 milliliter of 0.4% stock solution and 1 milliliter of seawater. Well 3 contained the 1 to 10 dilution, which was 1 milliliter of 0.4% stock solution and 9 milliliters of seawater. Well 4 contained the 1 to 100 dilution, which was 1 milliliter of 1 to 10 solution and 9 milliliters of seawater. Well 5 contained the 1 to 1000 dilution, which was 1 milliliter of 1 to 100 solution and 9 milliliters of seawater. Day 1 observations concluded that 60% of the shrimp died in the 1 to 1 ratio well, 50% died in the 1 to 10 ratio well, 40% died in the 1 to 100 ratio well, and 40% died in the 1 to 1000 ratio well. Day 2 observations revealed that 90% of the shrimp died in the 1 to 1 ratio well, 70% died in the 1 to 10 ratio well, 80% died in the 1 to 100 ratio well, and 70% died in the 1 to 1000 ratio well. Day 3 observations showed that 90% died in the 1 to 1 ratio well, 80% died in the 1 to 10 ratio well, 100% died in the 1 to 100 ratio well, and 100 died in the 1 to 1000 ratio well. The safest concentration of brine shrimp was the 1 to 10 concentration, which had 1 milliliter of stock solution and 9 milliliters of seawater. The safest concentration was determined by observing the number of living shrimp on day 3. Applications that could be used with humans include drug and gene delivery, the detection of proteins, the probing of DNA structure, tissue engineering, the separation and purification of biological molecules in cells, and MRI contrast enhancement.